All right. Today we're going to be talking about water, urea, and sodium reabsorption in the kidneys. We're primarily going to go through water and urea, but we're going to tagline it with sodium because that's a really important ion in this process. So first off, I want to give a quick overview of a nephron, which is the functional unit of a kidney. A functional unit is the smallest unit of an organ per se that can do all of the functions of that organ. So essentially this is a miniature kidney if you want to think of it that way. And there's two different components. There's the tubular component, which exits through this collecting duct. So pretty much all this sort of yellow thicker tube is the tubular component. And then there's the vascular or vein/capillary component where the capillaries surround these tubules. And in between these two spaces is a really important space called the interstitial space. And we'll see things diffusing across their gradients into the capillaries and into the tubules, depending on which ones we're talking about. Now, the goal of the nephron, or the kidneys, is to help reabsorb good materials, so materials that are beneficial to the body, like water, sodium, calcium ions, those types of things, excrete unwanted materials, that's like antibiotics that don't need to be used anymore, any metals or lead, those types of things, and maintain a material balance in the blood plasma so that you have normal blood pressure and that you've got enough materials to pretty much do all the functions of the body in the blood plasma so that they can readily be taken up by cells. Now, there's problems with this innately, is that we need some transporting mechanisms to bring things in and out at good capacities, and also we need to not have mechanisms to transport unwanted materials back into the body. So we're going to see a lot of channels that help bring these good materials back into the capillaries, and then we're also not going to see different passageways for those that aren't going to be used by the body again. So I just want to give a quick overview. So this is a glomerulus, and that's right here. So we're zooming in here. It is also called Bowman's capsule, which surrounds the glomerulus. And so if we zoom in here, this is where the filtrate will enter the tubular components of the nephrons. And as we can see here are the capillaries that are going to surround this tubular component, and here's the interstitial space where things will diffuse across. So what I want to overview here is that we've got sodium that's going to be actively transported out of the tubule, and we're going to have a gradient, electrostatic gradient that will actually, or a hydrostatic gradient that will move water back into the capillaries to be reabsorbed. And then as this filtrate decreases, we'll see urea actually going back into the capillaries as well, but we'll talk about the urea a little later. So let's start with water. So first off, in any reabsorption of any molecule, there's 5 different steps or 5 different barriers to overcome, and I'll just list those quickly. First, they have to leave the tubular fluid right here and cross the luminal membrane of the tubular cell. And then, so for water, the way it gets through there is through an aquaporin channel. So this is a water hole channel, so the water will go through that hole, and then it needs to pass through the cytosol of this tubular cell. So water will naturally move throughout this cell, and then it has to cross this basolateral, so the base to the side, which is the base to the side of the cell, to enter this interstitial fluid. So water, for example, will move again through aquaporin channels, but that is only facilitated by the active transport of sodium out of this tubular cell. And what that does is it increases the sodium concentration out here, which water will want to, fill in. So it wants to come to equilibrium. So water will be encouraged, essentially, to go out into the interstitial fluid, and then it must diffuse through there. And since these capillaries are only 1 cell thick, the water will easily diffuse back into the capillaries through that whole process. And so we pretty much went through all of this already, but just a couple of things to note is that we've talked about vasopressin in the past, and vasopressin increases these aquaporin channels both on the tubular side and the basolateral side of these tubular cells, which will increase water movement throughout, and it will be passively diffused. So water just passively diffuses through osmosis in those channels. And sodium ions are actively pumped, as we mentioned, and this requires ATP. We'll get to this later, but it actually uses 80% of the tubular cell's energy to actively pump these sodium molecules out. So we'll see how important that is later on. But I also want to talk about urea reabsorption. And now, urea is kind of an odd molecule. It's a waste product when we break down proteins so that we can use those individual amino acids to build back our muscles and build back our cells and so forth. But it's also a byproduct of ammonia, which is kind of, I want to say, toxic to the body in a way. So the hepatocytes in the liver will convert it to urea, which is a little less acidic, a little less dangerous molecule, and that will be freely floating throughout. And there's a couple things that urea helps with. I'll show on the next slide. But what I want to show is that urea is a bigger molecule. So urea won't just actively move across these channels as easy as like water and even sodium. But what happens is that up here we have 125 milliliters of filtrate. And as you can see, the urea concentration is moderate up here. But as we go down and as water and sodium move out, we're actually going to decrease the filtrate down to about 44 milliliters. Now, if we think about that, we're essentially decreasing the water volume of filtrate. So now we have a really high concentration of urea because the molecules of urea have stayed the same throughout. And now it's a really high concentration. It'll actually be passively diffused down its concentration gradient since it's slightly charged, and it can actually move across these membranes and then move back into the capillaries. And in the proximal tubule, about 50% of urea will be passed back into the blood vessels, but some will be lost as urine, which gives urine its characteristic smell of like ammonia. That's actually urea. So that's urea. But I do want to show how these two molecules kind of interact in the nephron in this loop of Henle. So the loop of Henle is on the, I think this is the medullary side of the tubules, medulla meaning lower, like the medulla is in the base of our brain. And so after this proximal tubule, so yes, it goes through the glomerulus, moves down here, and 50% is going to be reabsorbed for urea at this point. But we still have some urea floating around in this interstitial fluid. And when urea crosses back into the tubules in this descending loop of Henle, water will be almost forced out. So it's almost like an exchange program, we'll call it. And this is called the counter-current multiplier system. Essentially, counter going opposite ways in currents, and it's multiplying. So slowly as you go down, water is going to be coming out and being reabsorbed, and urea is going to be going back in. I don't know the specific pressures and how the osmotic gradients work here, but just know that these two things will kind of exchange. And like I said earlier, 50% will be absorbed for urea by about this point. And then we're kind of bringing back in urea into the tubule, but most of this urea is actually reabsorbed in the collecting duct at the end of the nephron. So if we go back to this screen, here's the collecting duct, which will actually go to your ureters and go back to the bladder eventually. But this is where urea will be reabsorbed in higher excess. And now this presentation is only going to be primarily about water and urea, but as I was researching, there's a lot of, I guess, important things that happen with sodium reabsorption into nephron. So I just wanted to briefly overview this. Now, there are three kind of main steps of sodium reabsorption, and most of it will happen in the proximal tubule. I think it's about 67%, where sodium will be reabsorbed into, and when I say reabsorption, I mean back into the capillaries, not back into the nephron, back into the capillaries. And so in the proximal tubule, sodium plays a huge, huge role in symports. And symports mean we're moving both molecules a certain direction. So say like sodium with glucose, sodium with amino acids, sodium with chloride ions. They will move with sodium as it's being actively pumped or as it's being moved to the interstitial fluid, to the capillaries. So it's almost aiding in moving several different materials, helpful materials that the body wants, back into the capillaries. A smaller amount will be reabsorbed in the ascending loop of Henle. And notice how I said ascending. As we look here, water is kind of being reabsorbed, and sodium and chloride are going to be dissolved into the interstitial fluid, into the capillaries again. And this will be adjusted based on fluid conservation needs. So say you're incredibly dehydrated, it will obviously raise your blood pressure, or I guess decrease your let me not get too confused on myself, but just based on how much water intake you've had, based on how much sodium intake you'd have, this will adjust in the ascending loop of Henle. So this is kind of the variable amount that will be reabsorbed. And then finally in the distal and collecting tubules, as we saw earlier. And this will be adjusted based on hormonal control. We've looked at aldosterone in the past about how this is going to bring potassium ions out of the bloodstream and putting back sodium ions in the bloodstream based on different osmolarities in the blood and based on blood pressure. So this will help regulate that extracellular fluid on the outside of the blood vessels to help maintain a good blood pressure number. So, and there's just a few other things with sodium. I mentioned this earlier that 80% of the tubular cells use their ATP for sodium transport. So 80% of the ATP they're using are to actively pump sodium, which is a huge deal, You hear about this a lot also in pacemaker cells and in nerve cells. There's a lot of ATP being used for that sodium-potassium pump. It's really, really important. I mentioned this earlier, that sodium is transporting different other organic molecules for reabsorption, the ones that are beneficial to the body. And this is just a staggering fact that 99.5% of sodium ions are reabsorbed in the body. So we maintain a very steady amount of sodium, which is clear that we need that in so many other tissues like your neuromuscular junctions and your pacemaker cells of your heart. So it's really important to keep these molecules circulating around your body to have them readily available. And also this active pumping into the basolateral side. So let's move this right over here. This is the basolateral side. It will keep the tubular cells' concentration of sodium very low, which if we think about it, say we bring in a bunch of organic molecules here. Okay, well, they won't be able to get back over this way. Diffusion will be wanting them to move towards the interstitial fluid because sodium is being actively transported into here, which we want to kind of put other molecules into this to break out all the sodium and to create equilibrium. So it's going to encourage other movement of materials into here. And it also establishes that hydrostatic pressure I mentioned earlier in this fluid to help push water back into the capillaries, which is also vital because, you know, our bodies need water here and there. Here are the references I used. A couple of them were online Lumen Learning textbooks, which are incredibly helpful, and then our textbook, Sherwood. So thanks for watching, and I hope you learned something new.